Cell membranes have many signal receptors in them that are vital to the functioning of the cell. The G protein receptor system is a series of chemical reactions that ends with a cellular response. G protein coupled receptors are a type of receptor present in cells and is activated when a ligand binds the G protein linked receptor. It is important to remember that not all chemical messengers fit in all receptors. They are specific to each receptor. When a signaling molecule binds to a receptor, it turns active and results in a conformational change in shape. The signaling interaction occurs between the ligand and the G protein coupled receptor. The G protein in the cell cytoplasm undergoes a change when a structural change occurs in the receptor. The inactive form of G protein is binded to the receptor, and guanosine diphosphate, also known as GDP, is also attached to the G protein. When the protein becomes activated after attaching to the signal receptor, GDP is made into GTP, which is guanosine triphosphate. This GTP activates the G protein. Next, the G protein that is activated diffuses along the plasma membrane, in which it heads towards an inactive enzyme. For this specific case, the enzyme is adenylyl cyclase. The G protein then activates the enzyme and returns to its original state. The enzyme that was activated by the G protein converts ATP to cyclic AMP, also known as CAMP. This is a secondary messenger that goes on to activate protein kinase, which phosphorylates other proteins, activating many more kinases. Then the cell reaches the end of the signal transduction pathway and then moves on to the cellular response. For this case, the response is calcium ions being released from the endoplasmic reticulum.
Next up is the tyrosine kinase receptor system. Receptors are outside the plasma membrane, and the tails of the tyrosine sits inside the cell cytoplasm. The signal protein binds to the receptor, which makes the two receptors move together, creating a dimer. ATP gives 1-phosphate to the tails of the tyrosine kinase, which causes it to become ADP instead. ADP is adenosine diphosphate. Now the receptor is completely ready to go. Inactive relay proteins then attach themselves to tyrosine kinase tails, and they alter their shape, becoming active. The activated relay proteins then activate the signal transduction pathway, which produces a cellular response. Again, in this case, the response is the calcium ions being released by the ER.
Anthrax poisoning. Anthrax poisoning affects the G protein receptor system. Anthrax poisoning ruins the G protein pathway. When inhaled, anthrax, which is an injurious bacteria, attaches to the human's lungs. From here, it continues to grow. As it grows, a toxin is released, which conforms the shape of the adenylyl cyclase, altering it from its proper functioning state. A ligand attaches to the receptor, as shown in the model. The GDP is continued to turn to GTP as the process continues. Now, the activated G protein attempts to attach to the adenylyl cyclase, as it would do if it were unaffected by the disease. However, it cannot perform this task, for its shape is different and will not attach properly. Due to this occurrence, ATP does not convert into CAMP or CAMP anymore. It cannot send messages, so the cell dies thereafter from its disabled state. This death happens because the cell cannot activate vital transduction pathways or carry out essential functions for its health anymore, which inevitably leads to human death after many cells die. Yikes, indeed. Symptoms of this disease include inflammation of the spinal cord and brain, coughing up blood, a high fever, nausea, shortness of breath, and more, even including death. However, do not fear, treatments are here. Treatments include antibiotics, which halt the bacteria growth, or antitoxins that target the toxins made by the bacteria, but can only be administered with special care and attention, and have a strong effect on the disease. Thank you for watching.